Hello and welcome to the special edition of To The Point. We have with us today Dr. Ramakrishna, who is quite well known in India, received the Nobel Prize in 2009 for chemistry. Dr. Ramakrishnan, good to have you with us.
Thank you.
Coming to the kind of work that you have done, for which, of course, you have been recognized with the Nobel Prize and so on, what is the current area of your work? Is it a continuation or are you working on something which are new?
It's a continuation. I'm slightly fortunate that translation is a very large, complex, and fundamentally important problem. And so the structure of the ribosome that we did in 2000 was only the beginning of a new stage of studying how the ribosome works, how it's regulated, how it's different between humans and bacteria, how viruses, for instance, will hijack the ribosome to get it to make their own proteins instead of the host cell's proteins. These are all big questions that still remain to be studied in detail. And so it's a topic that continues to have great interest in molecular biology. So in that sense, it's perhaps fortunate that I didn't have to then go around and looking for something else to do.
When you said translation, you're really talking about how finely proteins are created in the cell. That you have the process of DNA transcribing RNA and then, of course, the translation process where the actual proteins are created.
The reason it's called translation is very interesting because genes, both DNA and RNA, which is a sort of direct copy of a piece of the DNA, use a language of nucleic acids, which is a four-letter alphabet. There are four types of bases that go up to make DNA or RNA. And this string of bases specifies the sequence of amino acids to make a protein. The amino acids are the building blocks of protein, but there are at least 20 types of amino acids. There are actually a couple more, but there are 20 major types of amino acids. And so it's a question of going from a four-letter alphabet, a sentence in a four-letter alphabet, to constructing something that has a 20-letter alphabet. And that's why it's called translation. So you're using a code to translate from one sort of language to another.
So this is the protein language which finally is translated into from the DNA, the genetic information code that exists on the DNA.
Yes. And proteins are polymers of amino acids, and they fold up into specific shapes, and that then allows them to carry out their very specific functions. So proteins do all sorts of things in the cell. The only way you're able to look at me now or any of the viewers is because a protein in your eye called rhodopsin can sense light. And proteins carry oxygen in your blood. Proteins are antibodies that help to fight disease. So every protein carries out a specialized function, and each protein is made by the ribosome by translating information in a very specific gene that codes for that protein.
So ultimately, the hereditary information that goes from cell to cell, that's really the three steps: the DNA, the transcription, and the translation. That's really what is the final puzzle of life.
But it's much more than that. For instance, you have to ask, why do your skin cells not make rhodopsin, which is in your eyes? And why don't they make antibodies? And how come, your neuronal receptors are not made in your liver? So proteins are, the synthesis of proteins is highly regulated and highly specialized depending on the cell. The cell also controls when to make a protein and how much to make of it, when to destroy a protein. So it's a highly complex, regulated system. So the act of making the protein is just one part of a very complicated sort of control structure, which is what gives us life, if you like.
Dr. Ramakrishnan, fundamental advances in science quite often are important in themselves. They don't necessarily have immediate applications. But in this particular case, it seems that it has direct application in terms of how the antibiotics respond to, or how antibiotics really react to various cells. various parts of the bacteria and as well as the human cells. So do you think that this would really lead to immediate solutions?
It has good prospects. So what you alluded to, antibiotics, it turns out that a large number of antibiotics work by binding to the ribosomes of bacteria, but not binding quite so well to the ribosomes of humans. And this difference in binding allows us to use them as medicines, because you can have a dose where they affect bacterial ribosomes and kill off bacteria but don't kill us. Now, because you determine these high-resolution structures, you can now see exactly how these antibiotics are bound to the ribosome. These antibiotics were discovered, some of them were discovered 50 years before the structure of the ribosome, but no one knew precisely how they bound. Once you know precisely how they bind, then you can start thinking of designing compounds that might bind in that pocket even better, and that might bind to ribosomes that are resistant to that particular antibiotic because of a mutation in the binding pocket, so that the natural antibiotic doesn't fit properly in the pocket. But you could design something else that might fit regardless of whether it's resistant or not. So Tom Stites and Peter Moore and their colleagues, as you know, Tom Stites shared the Nobel Prize for the ribosome. They founded a company, a biotech company called Ribex Pharmaceuticals, whose purpose was to use these ribosome structures to design new compounds. And many of them entered phase 1 clinical trials. Some of them have gone through phase 1 clinical trials. So I think this sort of idea is very much in the making. But it turns out, there's a different problem with the pharmaceutical industry, and that is it takes a lot of money. Typically, the figure is about $1 billion to develop a new drug, going from basic science all the way to a medicine that you can sell. Okay, and so pharmaceutical companies have to invest this sort of money. They have to try and recapture it. Now, infectious diseases have a particular problem in that there are a large number of antibiotics, so a new antibiotic is usually reserved for bacteria that are resistant to the more readily available antibiotics. Okay, so that means it's a small pool of patients, because you don't want to give it to everybody, because then you'd have resistance to the new one, and then your $1 billion investment turns out again to be problematic. So that's a problem. So how do you recover that kind of money? The other problem is, with an antibiotic is you give it to a patient and a week or two later, they're often cured. Pharmaceutical companies would prefer something that the patient has to take the rest of their life, like blood pressure or cholesterol, medication. That's where the money is. And so I think a different sort of model needs to be created for development of these new.
We'll come back to this in the next segment that we are going to do. Time to take a break. Keep watching Rajya Sabha TV. Welcome back to The Point. We are having a discussion with Dr. Ramakrishnan on what could be a possible new model for pharmaceutical companies. Dr. Ramakrishnan, you said the current model, and you talked about $1 billion investment for pharmaceutical companies, which perhaps is a figure which could be questioned, but that apart, that we really need a different model of developing new medicines, because the current model based on the market is not working. What would be your idea of such a new model?
Well, I'm a laboratory scientist, not a businessman or an economist. So I hesitate to suggest alternatives. But I think one possibility is that perhaps governments or international health organizations could fund the development of new medicines. In other words, fund the process of taking something from basic science through clinical trials, through lead compounds and then clinical trials, to the point where they can become useful, and then perhaps license them to a variety of companies rather than to one proprietary product. And this might also help the problem with, say, developing countries or poorer countries where these new medicines might be too expensive for treatment. And in fact, infectious disease, as you know, is a bigger problem in countries like India or in Africa than in the West. Yes, there's infectious disease in the West, but it's a much bigger problem in some other parts of the world. And those are precisely the parts of the world where they cannot afford, brand new medicines created by Western pharmaceutical companies. This is not the fault of the pharmaceutical companies. They're, profit-making companies. They need a return on their investment. So it's pointless to blame pharmaceutical companies for, trying to recover their very large investment. I think perhaps if governments and international organizations could get together and somehow provide an alternative investment path, then that could help this process.
I think that's a very, very important point that you're making, that in fact the pharmaceutical companies, if they look at their own business model, their interest would be for medicines which you have to take lifelong. So that is the kind of diseases that they are interested in.
It biases them. There are a number of pharmaceutical companies that continue to develop antibiotics, okay? So it's not true that they are ignoring it. It's just that it's harder for them to, make money on their investment.
If there is a business model and all pharmaceutical companies are responsible for their business models, then there is an incentive for live medicines that you take lifelong, infectious diseases you get caught in 2 weeks. There's a less of return on investment, as you said, and without getting into whether it's as an ethical issue, purely as a business issue, there is a problem. So what you are saying is the states and international organizations should play a much bigger role in translating the developments of laboratory science into finally clinical trials and getting it out.
I think that's true. I would agree with that.
You must have heard about something that CSIR in India is doing regarding open source drug discovery. So if you have looked at it, how do you think that would work as a model?
I've only heard about it briefly. Okay. It's an interesting idea, but I don't know whether it'll actually work. I mean, I think you have to wait and see what the results are. And I have to say it's a long way from getting lead compounds and inhibitors to something that actually works. I learned this, I knew nothing about it, but I learned this through my involvement with Ribex and with an earlier company in Britain. So the difficulties of taking something that, say, inhibits the ribosome or any target, okay, to something that's useful as a medicine are quite large. You have to make sure that the compound is not toxic. You have to make sure it's absorbed in the cells, that it has a sufficient lifetime, that doesn't have any long-term effects. These are big problems, and that takes a dedicated group of people. It takes medicinal chemists, it takes people who know physiology, who know clinical work. So it's not an easy problem to tackle. So I'm not sure which approach is going to work.
And I think, again, what you're making is a very important point again, that when you do something in silico, in the lab, and then you want to take it to actual life forms, there's a whole set of issues involved.
Yeah, and in fact, it's sometimes the larger problem. Often you can get lots and lots of interesting lead compounds, but to distinguish between those and then try and find things that are high affinity, that are actually working, that's still a big problem. And in fact, if you look at the development of drugs, often most of the money is in that second part. Most of the expense is in that second part.
So one is a question of investment, one is a question of science. The second part is more at the moment empirical, more art than science, if you will.
Perhaps, but it's not entirely, but there's a process of how to test drugs, how to do clinical trials.
And that's a much longer process.
It's a longer process,
you were
and it's
saying
expensive.
That while one part of the problem is more amenable to a certain kind of analysis, this part still remains as a major cost and so on. But you do think that in this case, that the model has to change perhaps, particularly for infectious diseases and what are called the forgotten diseases. It has to change to a more publicly driven and more publicly funded research.
I really do believe that, yes.
And that's a very important point, particularly for countries like India, Africa, large parts of Asia, which are really suffering a very high load of infectious diseases. So you would say that one of your suggestions, recommendation would be the government of India really talk things about this in a bigger way, as well as international organizations.
But of course, you know, there are other approaches. There's preventive medicine, which is, you know, perhaps doesn't have quite the glamour value, but it's, I think, far more important. And there's also vaccine development, there's diagnostics. There are many, many aspects to treating disease. And I think development of new medicines is only one tool in the arsenal that people should think about.
Larger socioeconomic condition which addresses the health
even
as a
better
part.
nutrition.
Better nutrition. In fact, occupation, as you know, health really improved with jobs. That's employment is one of the things which really drove up the health statistics all over the developed countries. So that's very much a part of what we're saying. But limiting ourselves to infectious diseases and diseases as of now, this would be something that would be of interest. We'll be back with the next segment of To the Point. Keep watching Rajya Sabha TV. Welcome back to Rajya Sabha TV and this special edition of To the Point. Dr. Ramakrishnan, in this last segment, we'd like to discuss what is a scientist's responsibility to the people. Because after all, the scientists today practice research in a relatively high-cost laboratory setup. So it's not free. It's not like earlier days you could do it in your little laboratory you set up in your house. So do you think the scientists owe it to the people to explain what are the consequences of the research?
Oh, absolutely. I think most scientists are funded by taxpayers. And since I work in Britain, my attitude is that if a farmer in Yorkshire wants to know from me why some of his taxes should go to support my research, I should be able to explain it to him, okay? And I think the public are not fools. They can distinguish, you know, when science is important for its own sake, when it has immediate applications and so on. And they're quite broad-minded. If you look at almost any key invention today and trace it back, you'll find that at its root is some basic curiosity-driven science. Whether you take electricity, whether you take television, transistors, satellites. You know, satellites are based on Newton's laws of motion, which were discovered, you know, several hundred years ago. And, you know, satellite technology has transformed the world. It's improving. It can be used for agriculture, for weather forecast, all sorts of things, right? So I don't think it's a problem for scientists to explain to the public why what they're doing is worth funding. And if they cannot explain it, then they shouldn't be doing it, okay? So that is, and I think too many scientists sort of sit in their ivory tower and are unable to explain or unwilling even to engage with the public. And I think all scientists who are publicly funded have a responsibility to be able to tell people why it is that they're doing what they're doing.
Dr. Ramakrishnan, one of the basic problems in this model of scientists explaining to the people that institutional mechanisms by which the scientists can reach the people really do not exist in most of the countries. Television is no longer public broadcasting, so it's become much more private, again, driven by the needs of the shareholders. So do you think there is a missing link today by which what role earlier public broadcasting, public radio played, and by which scientists could really explain what they were doing is missing today?
Yeah, that's true. Commercial television has a problem that these segments are short, you know, they are dependent on the popularity of programs and so on. Scientists actually can make their subjects very interesting. Some of the most interesting programs I've watched, in fact, some of the most interesting programs my wife has watched, and she is a children's book writer and illustrator, so she's a better example than I am because I'm a practicing scientist. Some of the most interesting programs she's watched have been about science and nature. So it is possible to develop. I mean, what we do is actually explore the mysteries of nature, right? And then we ask how they can be used. All of these aspects are interesting. And there's no reason why they cannot be made interesting to a large public, because, we're all born as scientists. When you're a child, you're naturally a scientist. You want to know how the world works, why things are, why is the sky blue, why people die. Those are all scientific questions. And then as we get older, we somehow stop asking these questions. But intrinsically, at heart, we're all scientists. Scientists is nothing more than a curiosity about how the world works, how nature works. And it should be possible for scientists to, convey that excitement about their work to the public.
But again, Dr. Ramakrishnan, the problem is the scientists may not be the best people to communicate using this new medium. As long as the public lecture, you're prepared for it, you know what it is. When you have to do that in a 10, 15 minute, 12 minute YouTube video, then you really need a set of skills. And that may require an institution. In fact, nature does it very well, and so do certain other scientific institutions.
So there's a great role for science journalists, for instance, who might have a science background, but have a training in how to portray scientific ideas to the public. And it's a tricky balance to be both accurate and intelligible, accessible to the public. So you would say that's a difficult balance. And
you would say that in India, for instance, you have been coming here, at least you know what is here, that part of the science communication is lacking. And if that is so, it will not produce the next generation of scientists.
I can only tell you personally what my experience has been, with a few exceptions. And I have to say, the Hindu is one of them. But with a few exceptions, most Indian newspapers have sort of concentrated on the gossip about my life rather than about the details of the work and what motivates me as a scientist, what's interesting about the science. They're doing a disservice to the public, and they're also underestimating the public, because if they actually explain science in an interesting way, science is about the world we live in. Everyone would normally be interested in it if it's portrayed to them in an interesting way.
So more the page 3 view of scientists rather a view of scientists doing science is what the media tends to focus on. Last question. You have been interacting with Indian students of science. You have come here a number of times. What do you think of Indian science? We don't have too much time, so a short answer.
I think Indian science has undergone a big change, perhaps in the last 20 years. And science, of course, takes money, so it's not surprising that the advance in science also coincided with India's economic growth. And I would say many good young Indian scientists who go abroad for a PhD or for a postdoctoral fellowship are coming back and going to good institutes. And so I would say the outlook is promising. And the important thing is to give these young scientists independence, to give them good security in terms of science funding, so that they can compete. But I know very bright young Indian scientists who've returned to India, and they're trying to do very good science here. And so I'm optimistic, but cautiously optimistic, because it's very easy to destroy something and much harder to nurture something over the long term.
Thank you very much, Dr. Ramakrishnan. We have come to an end of our segment. This is all the time we have today. Keep watching Rajya Sabha TV. We'll be back with a special edition of To The Point with another distinguished guest in the future.